Hello students, in the previous class we have discussed about the modes of nutrition in bacteria. And in this class we are going to start with the new topic, that is reproduction in bacteria. So reproduction in bacteria can be divided into two types, and it is of two types. They are of two types. First one is asexual or vegetative reproduction, and the second one is sexual reproduction. In the asexual reproduction, there is no gamete formation, and there is no exchange of genetic material. In the sexual reproduction, there is gamete formation, gametes are formed, and there is exchange of genetic material between the gametes. And in the asexual reproduction, there are three types of asexual or vegetative reproductions. They are: 1. Binary fission. So in this one, bacteria divides into two cells. One bacteria divides into two cells. Now, in this binary fission, for example, E. coli. Let us take the E. coli. This is the cell wall and this is the cell membrane. This is the mesosome. And this is genetic material. In the binary fission, the bacteria replicates. For example, E. coli replicates in 20 minutes. It divides into two cells, one mother cell and one daughter cell. Now, before replication or at the time of replication or duplication, the genetic material attaches to the mesosome of plasma membrane. Now, first of all, the karyokinesis takes place, and this is the plasma membrane. Here, two mesosomes. That is, the nucleus is separating. So, there is no nucleus in these prokaryotes, so we can call it as nucleoid or genetic material. So, this genetic material is getting separated. So, we can call it as karyokinesis. That is, nucleus is getting divided. Genetic material is getting divided. So, there are two nucleus which are produced or duplicated. Now, in E. coli, the nucleoid or genetic material gets separated in 18 minutes, and this is genetic material. And the remaining 2 minutes for cell division. So overall, it takes 20 minutes of time for one duplication. That is, one cell forms one new and one old cell. So one is mother cell and one daughter cell. One mother, which is old cell, and one daughter cell, that is a new cell. This is binary fission. It is most common procedure or most common in bacteria. Number 2. Budding. And this is very rare. Binary fission is very common in bacteria, and budding can be very rarely seen. Next, the third one is endospore formation. This is also a asexual reproduction. We can say it as asexual reproduction, but it is not an asexual reproduction or vegetative reproduction. In this, no division or no division or new cell formation takes place. But here in the endospore formation, the cell undergoes a deep sleep, that is it gets into dormant condition. That is, here this is E. coli. Let us assume the E. coli. So this is the E. coli cell, and the cellular components such as cytoplasm, genetic material, some of the ribosomes, and all of the ribosomes, and first starch granules, these are accumulated to one side of the E. coli cell, and they are surrounded by a thick layer of membranes. So this helps in surviving of unfavorable conditions. This helps in survival of unfavorable conditions. What happens when favorable conditions return? Here, when favorable conditions return, this dormant endospore gets germinate and forms a new cell. Here, there is no old cell or new cell. The existing cell just wakes up. That's all. Here, it gets into sleep and it wakes up into a new cell. There is no reproduction or no formation of a new or a daughter cell in this type of reproduction. Okay, these are the asexual or vegetative types of reproduction which are performed by bacteria. And in the sexual reproduction, there are three types of sexual reproductions in the bacteria. They are, number one, transformation. Transformation is explained by Griffith. This is explained by Griffith. What is a transformation? The genetic transfer of genetic material. So, the transfer of genetic material from one bacterial cell, from one organism to the other organism, This is called as transformation, which is given by Griffith. Next, number two is transduction. So this is explained by or given by Lederberg and Ginder. So in this transduction, transfer of genetic material from an organism to the other organism can be done through bacteriophage. This is called as transduction. And number three is conjugation. This is of these three types of sexual reproductions, conjugation is a little bit complex, in which genetic material transfer is done from one organism to the other through a conjugation tube. In this conjugation process, these cells are designed, or cells can be said as F positive strain and F negative strain. And F stands for fertility factor. What is this fertility factor? Is if we draw the E. coli cell, if we draw an E. coli cell, there is a mesosome and there is a nucleoid present all over the cell. There are ribosomes and an extra organelle which is present in the bacterial body or bacterial cell is the extracircular DNA, which is also called as plasmid. This is called plasmid. The cells which are having or consisting this plasmid is called as F positive cells, and the cells lack this plasmid is called as F negative cells. F negative cells. So conjugation occurs between F positive and F negative strains. Let us take the two cells with F positive and F negative. So this is the plasmid present cell, that is F positive one, and this is F negative one. Now there is a small flap like structure. This is called as pili, and we can call it as sex pili. So these flap like structures, when come in contact between the two organisms, this forms a conjugation bridge. So we can call it as a conjugation tube or conjugation bridge, through which this plasmid can be donated to the F negative or F minus strain from F positive strain through a process called rolling circle replication. Rolling circle replication. In this one, if F positive one donates the plasmid to the F negative one, there would be no plasmid remaining in the F positive one. But here, through this process or through this mechanism, the plasmid donates its part, some of the part, to the F negative one through rolling circle replication, which makes F positive and F negative ones as positive strains by donating some of its plasmid to the F negative one. Okay, so both of the cells now having F positive and F positive, that is plasmid. Okay, so this is the conjugation process. Now this is also not a section. Let us see transformation, transduction and conjugation. In these three types of sexual reproductions, we can say these are all reproductions, but there is gamete formation. Okay, but there is no zygote formation. There is exchange of genetic material and there is gamete formation, that is plus and minus strains we are calling them. Now we can say these three are sexual reproductions because there is no zygote formation and there is no fusion of positive and that is male and female gametes. So these are all reproductions and we can call them as sexual, but here there is no zygote formation. There is only the exchange of genetic material from one organism to the other. Now after the conjugation, these cells F positive and F positive, so these F positive cells or F minus cells, they divide by binary fission. And again, after this sexual reproduction, they are taking the binary fission as the main division for the cell division. Okay, so this is the class about the reproduction in bacteria. In the next class, we will discuss about the cyanobacteria, which is a blue-green algae.